Process classes. So in this lecture we will be starting with IIT JAM Biotechnology, and here we will be starting with biochemistry, enzyme biochemistry. So in the previous lecture we have started this enzyme biochemistry and we have done some enzyme kinetics too, but in this lecture we will be starting with the enzyme inhibition. So first let's get started with the real definition of enzyme inhibition. So what is enzyme inhibition? Enzyme inhibition is the compounds that converted compounds. So some compounds that convert enzymes. There is an enzyme and there are some compounds. These are converting this active enzyme to the inactive substance. Now when they are inactive substances, they cannot, this enzymatic activity they loses, means they cannot catalyze any reaction, and also this enzyme inhibition also affects the rate of enzymatically catalyzed reaction. Rate of enzymatically catalyzed reaction. Okay. Now then these type of process is called as enzyme inhibition. Enzyme inhibition. And the substances which are actually responsible for these type of inactive compounds or inactive substances, or to make an enzyme inactive, are called enzyme inhibitors. Enzyme inhibitors. Now these enzyme inhibitions have two types. Enzyme inhibition these has two types on the basis of their specificity. First is specific inhibition. So by the term specific inhibition we can say the compounds of the ion which are very specific in their effect are the specific inhibition, like inhibiting only one enzyme. There are some specific enzyme, so there are some certain compounds which are very much specific for their effect. Now the second one, this is the first one, the second one is non-specific inhibition. Non-specific inhibition. Okay, so this non-specific inhibition is obviously these are the substances which are non-specific for their inhibiting enzyme. So they can inhibit many enzyme. Whereas in specific inhibition they are specific for inhibiting one enzyme. So this is the main difference of the specific inhibition and the non-specific inhibition. Now this non-specific inhibition, on the basis of their separation, there are some types of subclasses of the non-specific inhibition. Okay, so let's see. Now this non-specific enzyme, we can subdivide it into two classes. One is reversible, and another one is irreversible. So these are the two types of non-specific inhibitors. Now what is reversible inhibitors? Reversible inhibitors are those type of inhibitors which are loosely bound to the active site of the enzyme. Loosely bound with the active site of enzyme. Okay, so this is why what happens after certain time there is a, so what happens after some time, it actually dissociates with the from the active site of the enzyme, or rapidly from the active site of the enzyme. So now these reversible inhibiting enzyme or the non-specific inhibitor, that also can be divided into three parts. First is competitive inhibition, competitive inhibitors, rather I say inhibitor. Second one is non-competitive. This is the second one, this is the first one, non-competitive, and the third one is uncompetitive. Uncompetitive. These are the three types of reversible inhibitors, non-specific inhibitors. And these irreversible inhibitors, first of all, irreversible inhibitors are the inhibitors, they are covalently bonded with the they are bonded with the enzyme. Okay, so these we can't further subdivide it into many parts. The irreversible competitive inhibitor or non-specific inhibitor, that is actually these are forming like enzyme, and they are forming with the inhibitor and forming the, so this enzyme and inhibitor will form a enzyme inhibitor complex, that is EI complex. Okay, and that will not form again the active enzyme. So that will, because that is the formation that is bounded by the covalent bond with the enzyme and the inhibitor. Now so the difference between the reversible and irreversible inhibitor is only, that is the bond that is forming between the enzyme and the inhibitor. So here let's study about these subdivision of the reversible inhibitor, that is competitive inhibitor, non-competitive inhibitor, and also uncompetitive inhibitor. These are the three subclasses that is under the reversible and non-specific inhibitor. So what is competitive inhibitor? First is competitive inhibitor. So competitive inhibitor is an inhibition where there is a competition between what? Enzyme is fixed, but there is a competition between substrate and inhibitor, because in these type of cases these inhibitors that is having the competition with the substrate. But if we just increase the concentration of the substrate in the solution or in this reaction, then the substrate will only bind to the enzyme. But otherwise there is a very much competition between the inhibitor and the substrate. But why there is a competition between inhibitor and the substrate? Because these type of inhibitors having a closely resemble with the substrate. So that is the competitive or the substrate analog inhibitor. That is why we call it as substrate analog. They look like substrate, and they always go and bound to the active site of the enzyme. Active site of the enzyme. So this is all about the competitive inhibitors. So that will form the either the ES or EI. Okay, so this is the competitive inhibitor. Now the next subdivision was the non-competitive inhibitor. The second one is non-competitive inhibitor. So by the name non-competitive inhibitor, we can say there is a no competition between the inhibitor and the substrate to bind with the enzyme. Why not? Because there is no such resemble with the substrate. The structural analog, they are not these inhibitors are not the analog of the substrate. Okay, so the formation is mainly like when there is a enzyme, there is a substrate, and they can also, this enzyme can bind with the inhibitor. And another thing, that is why they are not competing with the substrate, because these inhibitors that binds to the allosteric site of the enzyme, allosteric site of the enzyme. This allosteric site, actually the other site of the enzyme, that is not the active site of the enzyme where the substrate goes and bind. There are actually, if this is the enzyme, suppose there is this structure of the enzyme, this is the structure of the enzyme. So the substrate is, suppose this, and this is the active site, and this is the allosteric site. Okay, and suppose this is the inhibitor that binds to the enzyme allosteric site. So this will bind here, and substrate will go here and bind to the enzyme. So this is how this enzyme substrate inhibitor complex will form. So they are forming like EI, and also they are forming this ES, and then there is a binding of I inhibitor that will form ESI, and also there is a boundation like this, ESI. So there are the different ways how this ESI complex can be formed. Also from this ES complex, the formation of EP can process, that is the enzyme and the product. So this is all about the non-competitive inhibition, but also you have to remember that in as just like not like the competitive inhibition, there was this if you increases the substrate concentration, there can be the overcome of this process. But here if you just increase the substrate concentration, we cannot overcome the process of the non-competitive inhibitor, because there is always another side, that is the allosteric side, where actually they are making the boundation with the enzyme. Okay, so this was the competitive and the non-competitive inhibition. The third one is uncompetitive inhibition. So in the uncompetitive inhibition, there is a similarity like the non-competitive inhibition, but there are some differences obviously. Okay, so this is uncompetitive inhibition. Now in uncompetitive inhibition, what happened, that there is suppose the enzyme, and that is binding with the substrate first, and after forming the ES complex, then there is a addition of the inhibitor, so that will form ESI complex, and also from here we can get E and P, enzyme and product separately. So in the non-competitive inhibition, what I have seen, that inhibitor can directly also bind to the enzyme first, but here in uncompetitive inhibition also the substrate inhibitor go and bind to the allosteric site of the enzyme, allosteric site of the enzyme, like non-competitive inhibitor, but after forming the ES complex then only the inhibitor get bind to the ES complex and form this ESI complex. Okay, so there is no competition with the substrate that who will go first and sit with the enzyme. So first enzyme substrate complex will form and then there will be the inhibitor that will go and bind to the ES complex. Okay, this is how ES complex uncompetitive inhibition in the specific or the in the reversible inhibition take place. So these were the three types of inhibition, three type of reversible inhibition that takes place in the competitive inhibition. Okay, these are the competitive, uncompetitive and non-competitive inhibitors. So there are three types of reversible inhibition. Okay, now there are one thing that I haven't told you. I have told you about the irreversible inhibition. There is a covalent bond in irreversible inhibition. I have told you there is a covalent bond, so there is no coming back to the for their own formation, their beginning formation. Covalent bond. So on the based of these part, there is a inhibitor called suicide inhibitors. Suicide inhibitor. So this is a special class of irreversible inhibitors. These are the compound which are relatively unreactive. Now they are unreactive, but until they bind to the active site of the enzyme, until they bound to the active site of the enzyme. So just like competitive inhibition, they also bound with the active site of the enzyme. Okay, but they just possess or they just be unreactive. They are not reactive until they bound to the active site of the enzyme. Okay, so now they carry out the first few chemical steps. And like that of normal enzyme reaction, but instead of forming normal product, the inhibitor is converted to a very reactive compound. The inhibitor will convert it to a reactive compound. That combines irreversibly with the enzyme, that thereby inactivating it. That will combine to the, and this is the reactive compound that will form after the forming normal product. Okay, so inhibitor will be the reactive product. Okay, so after converted into a very reactive compound, that will combine with the irreversibly with the enzyme, enzyme. So then let's have a look what is happening here. So first, they are unreactive. They are unreactive. But when they are getting reactive, when there is an enzyme having the active site, they are bounding with the active site, this type of inhibitor. Okay, so they become reactive. So these inhibitors are getting reactive compound. So this is become a reactive compound, and they are forming this irreversible compound. So they are no going back to the enzyme part or the normal previous part. Okay, so this is we call as suicide inhibitors. Okay, because they utilize the normal enzymatic reaction mechanism to inactivate enzyme. Okay, these are the enzyme inhibitors. So we have now the idea of what is enzyme inhibition and the compounds that are responsible for the enzyme inhibition, or which are actually inhibiting the enzymatically reactions. They are called, these products are called enzyme inhibitors, and we have subdivided them via flowchart to many subdivision. Like first one is reversible and irreversible, and there are first one is specific and non-specific. Going to the first page. So this is the flowchart. First there is specific inhibitor, non-specific inhibitor. Then what is the difference between specific and non-specific, I have told you. Then what is the subdivision of the non-specific inhibitors, which have the ability to inhibit many enzymes, not the one enzyme. So they can be of two types. First is reversible, second one is irreversible, and we can further subdivided irreversible inhibitors, or we can subcategorize it into suicide inhibitor. Suicide inhibitor. And this reversible inhibitor, that is also subdivided, we can subdivide it into three parts, that is competitive inhibitor, non-competitive inhibitor and uncompetitive inhibitor. And also we have little details about these all the classes and the subclasses. So thank you for watching this video.